Good afternoon. I'm Jan Witkowski. I am Director of the Banbury Centre here at Cold Spring Harbour, and this is the first afternoon of the 80th Cold Spring Harbour Symposium on Quantitative Biology. And the title this year is 21st Century Genetics, Genes at Work. And I have with me David Kingsley from Stanford University. In Bruce's Stillman's opening remarks, David, he made reference to Mendel, because of course this is 150 years since Mendel's work. But there's another 19th century scientist who I think will be very interested in this symposium, and particularly your presentation, of course, Charles Darwin. You haven't spoken yet. Can you give us just a brief rundown on what you're going to talk about on Saturday?
Sure, and it's a very fun meeting for me to come to because the work we've done is focused on trying to use Mendelian principles to study evolution. So those two figures that you mentioned are icons for our own interests. Darwin didn't have a hereditary mechanism, and a lot of the biggest holes in the origin of species were a lack of information about where variants came from, how they segregated in families, how traits could disappear for a while and then reappear. And Mendel fixed a lot of that. He fixed it in papers published only 5 or 6 years after Darwin's work, but Darwin really never knew about it. And so a poignant history, I think, actually. And if you look at research today, you can revisit the same questions that Darwin and other people worried about, but using Mendelian principles to try to figure out where are the key chromosome regions that control evolutionary change. What are the actual genes that underlie the interesting traits that we see between natural species? And we've had a lot of fun looking for systems that would make it possible to study naturally occurring species by genetics. And you can do that. You have to look for loopholes in the species definition.
Presumably it's very important what species you choose to study.
Right. So that's the paradox, right? I mean, the definition of species is they're reproductively isolated. And you would think that puts the origin of species outside the realm of traditional genetics. About 15 years ago, we decided that to answer lots of questions in evolutionary biology, we would have to take an unbiased genetic approach to try to figure out what had happened in naturally occurring animals. So we went looking for organisms that had evolved recently enough that you could still cross them. And there are loopholes, as I said, in the species definition. You can be reproductively isolated for lots of different reasons, including inviability or sterility. But that's sort of the end game of species. Earlier than that, you may have separate species because they don't want to mate with each other. There may be behavioral incompatibilities or mechanical incompatibilities. Those can be overcome using artificial fertilization in the laboratory. So that's what we went looking for, was recently evolved things, natural species, but evolved so recently that it was really only behavioral and mechanical incompatibilities that were keeping them apart. And we found that in these young fish called the three-spined stickleback, which radiated just in the last 10,000 years. They're all over the place. There are a lot of them out there in the bay. And they show remarkable differences, morphological, physiological, behavioral. But you can squeeze out eggs and sperm from these very different looking fish and set up crosses to try to map the genetic basis of evolutionary change.
So these very different looking fish are isolated in some form, or is it simply, as you said, the fact that they don't want to mate with each other that's leading to isolation?
There's tens of thousands of different stickleback populations that got set up by the process of marine ancestors colonizing and giving rise to new freshwater populations when the glaciers melted. If you look at that whole range of populations, there's a spectrum of what I would call biological incompatibility. There are pairs that if you put them together, they're sterile. But a lot of them, if you put them together, they just don't want to mate with each other. And those can be overcome by setting up fertilization in the lab.
So what sort of traits? So you now have one species that's that's variable. What sort of traits have you been looking at?
That was part of what attracted us to them. They, despite it only being 10,000 years of evolution, they show huge differences. So 30-fold differences in the number of armor plates along the side of the fish, doubling of the number of teeth. Some of the populations that adapt to different food sources have completely transformed their jaw and their chewing apparatus to take advantage of different things. Presence or absence of entire fins. So fish are like land animals. You're supposed to have two sets of paired appendages. And in a lot of the stickleback populations, the hind fin has completely disappeared. Same kind of trait that you see in whale evolution.
Yeah.
But here studyable in a fish that evolved it just in the last 10,000 years, and you can still cross them.
Sticking with the evolutionary side for just a moment before getting onto the genetics. So are these sticklebacks peculiarly variable as compared with other organisms?
That's an interesting question. I think of them as just simply one of the most recent and dramatic examples that because they have this migratory life cycle that set things off just when the glaciers melted, we have an opportunity to see dramatic evolutionary change very recently. But I think the kinds of things they do are a microcosm of what's played out over and over again in the history of life on Earth.
Okay, back to the genetics,
Yeah.
the genetic side of the
Well,
equation.
I had interesting discussions when we were going to start this project because there were a lot of people who felt that evolution was really complicated. And so complicated that if you tried to set up genetic crosses between very different naturally occurring species, the differences would be due to thousands of little tiny changes that had all been accumulated over time to make the differences that you see today. And Darwin actually believed that. You know, he said over and over again that nature doesn't make leaps,
Yes.
that evolution is going to be based on infinitesimal changes that are accumulated. And when Mendel's laws were rediscovered, the neo-Darwinian synthesis uses Mendelian principles, but it takes the effect size and makes it, again, almost infinitesimally small.
Right.
So what several colleagues said when we started was, it's never going to work. You know, you will not be able to find chromosome regions that have very large genetic effects because all of the effects that underlie evolution are these tiny little things that have been accumulated. That's not what we found. We found that if you simply cross naturally occurring species, that very often when we did the genetic mapping, we could find chromosome regions that controlled a lot of the trait. It wasn't Mendelian, right? We didn't find chromosome regions that could explain 100% of the variance in a trait. But we would find chromosome regions that explained a half or 2/3 or 3/4 of the variation in armor or teeth or presence or absence of a hind fin. And that meant, as a geneticist, there was a handle that you could then chase down to try to find the molecular mechanisms that had produced these dramatic morphological differences.
And so, what would be a good case to take as an example? The plating, the scales,
Or the fins. I like the hind fin loss because it's a trait directly similar to a trait that most people are familiar with. Whales evolved from land animals in the course of going to streamlined ocean swimming. They lost their hind fin, hind leg. I go back and forth between fish and land animals because the problems are virtually the same.
Right. So is the so you presumably found a gene that accounts for a large proportion of this
Yep.
of this loss of
Right.
reduction.
So if you map pelvic reduction, you get a major locus that does about 2/3 of the variation in the trait, and 4 or 5 what we call modifier regions that each might control a few percent of the variance in the trait. The major gene, that major chromosome region, is a very well-known developmental regulator, a master transcription factor called PITX1. That's a homeodomain transcription factor that binds to and regulates all sorts of genes during normal development. The name actually doesn't sound like it has anything to do with hind limb or pelvic development at all, and that's because, like many of these key developmental regulators, it does a lot of other things during normal development, and it's required for normal pituitary formation, which again, it's directly related to a classic debate in evolutionary biology, which is the idea of monsters. Right?
I was going to come back to who was the hopeful monster?
Yeah, well, Goldschmidt proposed hopeful
Yes.
monsters, and the leaders of the neo-Darwinian synthesis, absolutely hated that, because Goldschmidt was saying the interesting differences might have emerged all at once in a hopeful monster, and the counterpoint was genetic changes that do big things to animals usually are deleterious.
Right. So
They're hopeless monsters,
yes.
in the words of Mayer.
And I was going to mention Bateson's semiotic mutants,
Uh-huh.
but they would fall into this hopeless monsters where you suddenly get an antenna coming out.
Yeah, that's one of the things. So Goldschmidt pointed to the existence of homeotic mutations and gave those as examples of look at the remarkable things that can happen to an animal all at once. Mayer's response was the homeotic mutations are sterile and have reduced viability. It's hopeless. It's not hopeful at all. And to me, that was one of the real pleasures in finding a homeodomain transcription factor underlying pelvic reduction in sticklebacks, because that gene, if you knock it out, you get a hopeless monster. It has small hind limbs, but it dies at birth. It's got craniofacial abnormalities, cleft palate, pituitary defects, and it doesn't look like a very promising basis to evolve traits that have been under positive selection in natural populations. But nonetheless, that's exactly the gene that nature has used.
I mean, there were talks this morning about cells' tissue specificity of enhancer activity as a accounting for specificity of gene expression.
Yep.
So in this case, is PITX, why does it have such a dramatic effect on pelvis
Well, so
as opposed to other?
the way nature has made an advantageous change instead of a hopeless monster is really relying on exactly what you're saying, which is that these powerful developmental control genes are themselves regulated by highly tissue-specific enhancers that recruit their expression at different times and places during development independently of each other. So when we went looking at what was the difference between a natural species that had lost its pelvis through PITX1 or the marine ancestor, most of what PITX1 does is completely intact in the natural species. The protein region's fine, the expression in the pituitary is fine, the expression in the head's fine. It's lost a non-coding bit of DNA far away from the transcript itself that causes that gene to turn on specifically in the developing pelvis. And if you lose that information, you can get a huge hopeful monster effect at that place in the body and preserve everything else the gene does.
So when you knock out the gene, as you were saying earlier, and you get these, the
Hopeless.
the sterile,
Helpless. Yeah,
helpless
it's a
one,
disaster.
It's because you've lost everything.
That's right. Yeah. So nature essentially is making regulatory changes instead of coding changes, and that allows it to tweak specific aspects of gene function instead of globally everything that the gene does.
Yeah. Is PITX involved in the whale, in the loss of real
that's actually
limbs in tetrapods?
That's something we're actually quite interested in. There's a morphological signature of losing the pelvis through PITX1, which we've seen in sticklebacks, and you can also see in knockout mice. There's a funny directional asymmetry. So PITX1 is closely related to a cousin gene named PITX2. PITX2 is actually involved in left-right axis formation. So if you lose the pelvis by the PITX1 gene, usually the morphological effect is more severe on the right-hand side than the left-hand side, because the PITX2 cousin is still around on the left-hand side. So when the PIDX1 was knocked out in mice, we shrank the pelvis, but it was more reduction on the right than the left. Sticklebacks, if you do the crosses between the naturally occurring forms and map asymmetry, you get the PIDX1 locus. It's exactly the same direction. It's bigger on the left than the right. So we've only been able to address your mammal question so far morphologically, but we've been looking at the pelvic rudiments of whales and manatees. And at least for the manatees, the other mammalian example of pelvic reduction, they show exactly the same kind of left biased, bigger on the left than the right that we see in the sticklebacks and in the mice. So we are now quite actively trying to track down the regulatory regions in the mammalian gene that control pelvic expression, because the morphological signature is strongly suggestive that the same mechanism might be involved. It's turned out it's painful because after all the work to check down the pelvic enhancer and the stickleback gene, the sequence of that does not align to mammals. So we have had to research through the mammalian gene to find the relevant regulatory switches again.
Well, as a quick aside, the first proper director of Coastal Harbour, Charles Davenport, died after getting pneumonia
Chasing a whale?
dissecting a whale out on the east end of the island.
I will be careful during these studies.
There's something else I was going to follow up. So coming back to your sticklebacks out in the wild, are these various forms, presumably the ones that don't breed, would now be considered different species?
Yep, absolutely. And there are, for example, species pairs even in the same lake, where one form has specialized for the shallow water, one is specialized for the open water. Those forms have different sizes, different colors, different teeth, different armor, they eat different food, they encounter different predators. And if you follow genetic markers in the lake, they are, by and large, reproductively isolated. So they would meet the formal species definition.
And if you compared their genomes,
Yep.
the genomes presumably would be very, very, very similar. More similar than us and chimps, for example.
Yeah, that's right, they are.
So it could be just...
Maybe one base pair difference every 300 bases in the genome.
Yeah. And the number of genes involved in making these sticklebacks so different is sort of a handful.
Well, so that's
the major morphological ones.
You know, I actually think that's when I say it's possible to find the chromosome regions that control evolutionary traits, I don't want to minimize the complexity of the problem, because in fact we've done crosses between different stickleback forms and measured everything we can think of. And there's lots more things we haven't thought of. But we've probably mapped over 200 chromosome regions that contribute to some aspect of the difference between forms. And those are just the ones that are big enough for us to see, and just the things we've decided to measure. So the global issue of evolving a new form does involve a whole bunch of genetic changes. It's just, if you focus on a particular trait at a time, if you atomize the problem and look at the components of the phenotypes, you can track down the genetic basis of traits much more easily than would have been predicted by the old debates about infinitesimal change.
Okay, we should stop. David, thank you very much. But what I will do is, when I talk to Svante Paavo on Saturday, I shall ask him about doing this sort of work with Neanderthals and modern humans.
Well, okay, so let me say one other thing, because on Saturday, one of the other themes I'm going to talk about is we are trying to take the lessons that we've learned from how evolution works in sticklebacks and apply it to a search for the molecular basis of becoming human, right? And so that's an ambitious problem that faces a lot of exactly the same objections I remember hearing when we started the sticklebacks, which is, I'll bet that's really complicated.
Okay. Well, and good luck with it.
Right. And in fact, you can find signatures that we think are contributing to limb modifications, brain changes, other morphological traits, and I think human evolution is going to be a studyable problem as well.
Great. David, thank you very much.
Okay.